Thanks for coming, guys. We got a great show lined up for you today. If you'd like to support this channel, there's PayPal and Patreon links in the info box below. Like I said, we got a great show, all-star panel, and before we get to them, I'd like to shout out my Patreons, without whom this channel and show would not exist. Sam Hine, Felix Hung, Qatar Craig, Sally Bayless, Chow Yun Cat, Tina Baker, James Mays, Eagle Plane and Anchor, Silver Umbrella, and Texas Mike. Make no doubt about it, without their support, folks, this show would not be here. All right, get ready to go on Fairytale Evolution, Part 4. And since Adam's here, there'll probably be not only Part 5, but Part 6 and Part 7 by the end of today's show.
I've promised to be quiet, haven't I? Not just start throwing in too many random factoids that generate another disproof.
No. It was good. I'm sure that we can add loads of sections on here, but you made some, you had some good ideas last week, and I had to acquiesce because we're going to have to hit junk DNA, which is a joke. And in the end, functional sequence specified complexity. That's very important to wind everything up. So yeah, thanks for that, Adam. More work, but we needed to do it. So who we have here today is the beautiful Betty Van Velsen. How's it going, Betty?
Yeah, I'm a little bit sore throaty, but it's good.
Yeah, that seems to be...
Hope that I can do a good job at shouting out super chats.
That seems to be
You
running...
already got one.
That seems to be running around because the distinguished and honorable Nathan Oakley is under the weather too, so we're going to miss him today. I think he's listening in, but I would have liked to have him on the panel for Nylonase, right? But hey, that's the way it goes. Who else we got on the panel?
I'm here.
10th.
Yeah.
What's up?
You arrived.
So we got 10th man here. How's it going, 10th?
I'm doing fantastic.
You ready for the Evo game?
I'm ready for the Evo because I don't have a Mitsubishi anymore.
Yeah, right. And we have, of course, Adam, who already spoke up. How you doing, sir?
All good, my friend, all good. Evening, Ted. And I've not said evening to Paula yet, as you're about to, so evening, Paula, as well.
Good evening, Adam.
Good evening.
How's it going there, Paula?
Hey, Paula.
Going all right. Keeping busy. Keeps me out of trouble, sort of. A little.
Speaking of keeping things out of trouble, Anthony.
He says he's lost his internet.
How did he tell you?
In the chat somehow, I guess by a phone. You know, you can always do that by phone.
Yeah.
You need internet always, right?
Yeah, he was having some internet trouble beforehand, and I'm sure it's attached to the soccer games that are on today. Am I mistaken there, Adam?
Well, you know, I'm doing the same multitasking. I'm streaming and just keeping one eye on the Champions League a little bit. But I think he is genuinely having a few glitchy problems, so maybe he's had to drop this just to maintain his stream for the football. But he said he'd come in on his phone, didn't he, if it was a problem. So
he sacrificed being on the show for a game?
This is Scousers their football, mate, Theo, unfortunately.
It's
a deep ingrained religion.
And that's the excuse?
Well, we all have our religions.
Yes, we do.
It's his goal in life, QE.
It's his goal. Oh my God, here we go. Oh man, this is going to be a long show.
Well, let me shout out a super chat then from James Myers, and he says, "What's up, John?" And he says, "Hello, Betty." So that's good.
What's up, James? I already shouted you out, man. Hey, check your messages, James, and I'll get back with you. All right, so Fairytale Evolution Part 4. We want to get on with this or maybe a little bit more chit chat? We can start rolling. It's up to you guys.
Well, maybe before you dive in, just where are we up to? We're up to
Oh, yeah, I can review what we're
dispelling some of the statistics for me. This bit is really just about putting into context some of the numbers that are thrown around to support the premise that we've evolved.
Evolved? What's that?
Among
you said evolved. What's that?
Yeah, to support the premise we've evolved from a monkey. Exactly this process that apparently there is this progression of stages that demonstrate this theory.
Theory.
Yeah, I'm struggling now. You see, I was pulling there.
Yeah. I do that to everyone. And those people usually harbor a hateful grudge towards QE after it. So the reason why they do that or harbor that hateful grudge is because it's frustrating when I ask them to define it and give me the scientific theory for it, because it ain't going to happen. So what they try to do from the beginning, they just bypass what it is and start giving you evidence for it. Let me tell you, folks, before you start giving evidence for something, you have to define that something. And if it's a scientific theory, then you have to give the scientific theory first. Okay? Just to let you know, because if you don't define the thing, then you could just make up anything and say, yep, that's what it is. No. You have to define the thing you're providing evidence for first, right? Then give the evidence. That's the order of things. And I put up with this, and I shouldn't have put up with it for a good 3 years, right? Until I said, what the heck am I doing? I'm chasing my tail, going round and round every time I go on these forums, because that's all I do. I was battling retard Evos daily on the science or so-called science forums. And every day it'd be something new. They'd bring it up, or they'd wait a couple days and bring back the same thing up that I pounded a few days ago, bring it back up all over again, right? And we'd just go round and round. So I did that for a long time, and I shouldn't have. It was my fault. Until I said, okay, that's enough. Give me the scientific theory of evolution. That's all I had to say, because it was over then. They can't do it. So what are we talking about? Well, we're not talking about anything. And that's what I like to preface this today with. Number one, there is no scientific theory of evolution. So this is a caveat. There is no science in this whatsoever. Every topic we're going to talk about today, there is no scientific evidence. So there's no science in this one whatsoever. Number one, everything is begging the question. And I just refused to do it until I was badgered and pawed at incessantly over the past month or so to go ahead, John, just go ahead and beg the question. Let's go through some of these so-called evidences that they have, because people are getting bombarded with these, and let's go ahead and put out the arguments against them. So I just want to caveat this whole thing with those two statements. Any questions so far?
You were pawed at?
I was incessantly pawed at.
Oh, poor baby.
Yeah, that's right. That's right. And I shall not be pawed at. It's one of my refrigerator rules.
Refrigerator rules. That's where their cat ended up in.
Yeah. So the topics we're going to be going over today will be the 98 or 99% similarity with chimps. That was the actual claim. Of course, everyone says 98%. We're going to go over human chromosome 2 fusion. Those will be 2 quick topics. You'll know why they're quick when we go over them, because there's absolutely nothing there. Then we'll go over, Captain, there be whales here. Whale evolution, which is a flippin' joke. Then my ultimate favorite, Lenski's E.coli. And then we'll finish it up with Nylonase. And that'll be part 4 for today. Any questions on those? Any preemptive strikes? Feel free.
We end with nylons?
With nylon, yes, nylon.
Okay.
We have bacteria
Interesting.
that can break down nylon. So that's a man-made synthetic, right? But that's not the that's not the whole story. That's another typical tactic, right? All they do is throw out these titles. But it's just like with anything else, behind a title, there's got to be some type of coherent story or scientific evidence by it. You can't just throw out a title. What's your proof of evolution? Lenski's E. coli. That's not evidence. You have to know what you're talking about. You're going to see that these people are clueless. Absolutely clueless. So like I said, any preemptive strikes before we begin?
No, let's begin.
Let's take it to him.
Anthony, you here? That's what I thought.
I've just got one, John.
Go ahead, sir.
It relates. I don't want to go into specifics. It relates to the Nylonase, but as it relates to the Moreland, it's. As I said, I'd only looked into it this afternoon just because I said I'd wait for Nathan, and when I found out he weren't coming, I thought I'll have a little read. And again, it just goes into showing that it's a misrepresentation at best of the facts, because as soon as you do look into it, this supposed evolution that's occurred is nothing more than a very minor adaptation, and it's not some, in the case of the Nylonase, some strange thing that's evolved into this. It's a random thing that had this thing already, and when it encounters this product, it's a lucky coincidence that its quirky little function coincides with things. You know what I mean? And it's a misrepresentation all the time of the truth to support a narrative, and in most cases, it's done by omitting some of the more significant facts.
Did you say omitting?
Yes, omitting, as in not including the detail.
Yeah, you think?
Yes, just a touch.
They just give the title, right? Most people that are out there with these proofs, they're just parroting it from Wiki, right? No depth. You're going to get jacked up. I'm going to tell you, you come half step in this direction, you're going to get jacked up and jacked up quick. Just to let you know. If you don't know it by now, you should. All right, let's roll. What do you think? So we'll start off with, see, I put 98% similarity with chimps, but it wasn't 98, it was always 99%. I'm not being pedantic about it, but the actual claim was 99%, not 98. But I'd always get hit up with, well, chimps are 98% similar to us. Right? No. From Cohen, Relative Differences, the myth of the 1% in science. The first 90s, like I said, the first 99% similar came about by reassociation kinetics and extrapolations thereof. This was the initial technique. The 99% fiasco has many flaws, including throwing out data. It was put forward by Alan Wilson and Mary Claire King in 1975. Right? So this was a joke. All they're doing is comparing. This isn't science, folks. Anyone can compare something and then make up a just so story after it. Then the 97% fiasco from Libley and Ahlquist in 1987 from the Journal of Molecular Evolution, whatever that is. Still no 98% was conjured from DNA hybridization in the 80s, along with Walk Like an Egyptian from the Bangles. Shout out to Susan. So this was just all comparative jokes. And from Sarek et al. In 1989, DNA hybridization is somewhat arbitrary and clunky. Yeah, it's all arbitrary because you're making it up. Here is the death knell to all this 98-99% similarity junk. You ready? From Ebersberger, Mapping Human Genetic Ancestry, Molecular Biology and Evolution, whatever that is, in 2007. Listen closely. For about 23% of our genome, we share no immediate genetic ancestry with our closest living relative, the chimpanzee. Uh-oh, down to 77% right quick. That equals 690 million base pairs, roughly 227 large encyclopedias. And bear in mind, even if humans were only 4% different, this still amounts to 120 million base pairs, equivalent to approximately 12 million words or 40 large encyclopedias of information. It's really just over right there. Let's just nail it to the wall. Professor David Page of the Whitehead Institute of Biomedical Research, the Y chromosome in nature. The Y chromosomes of chimps and humans are horrendously different from each other. Now, I don't know how much different that is, but horrendously is probably a pretty big number. You think? We're not going to stop right there. We'll just keep on rolling because that's, you know what? Let's stop right there. What do you guys think about that? We'll stop.
Well, I just had a thought when you were talking about the 98%, so it leaves 2%. That's a big 2% since I don't see any chimps making any 747s or building bridges.
Just wait.
Yeah, it's the differences. Yeah, it's the differences that matter. I mean, if all you look at, I've heard that it's like 50% that we share with bananas, which is why some people are half bananas. Yeah, I mean,
excuse me. As somebody who partly identifies as banana, I find that offensive.
That's racist.
Well, you don't have to split at him.
Oh no.
Oh my goodness. Here he goes.
Yeah, I slipped up there giving him that.
Oh my God. It never ends now.
That's an appeal to something.
Oh my God.
Yeah, similarities. It's the differences. If you have, like, people are watching the chat a little bit. It's like, well, if it has a face and we have a face, then we must have an ancestor. That's just begging the question at the very kindest thing to say, because it's similarities only. If you have enough similarities, then there must be a connection. And they've, the statements in the chat, it must be possible. It must be possible. That's where they start. They don't start with empirical evidence, scientific method, anything like that. It's always fairy tale first.
Did somebody in the chat define the scientific theory? Did they post the scientific theory of evolution yet?
Nope.
That's what I thought. Adam? Who's winning? Anthony, I already know you're gone. Gotcha. 10th? You already gave yours. Betty?
Yeah, I've seen.
Oh, God, I'm unmuted. Sorry.
Oh, you were on mute? Okay, yeah. Go ahead. Sorry.
I said Betty. It was 2-0. It's 2-0 to Liverpool. I apologize.
I figured.
Yeah, sorry, Betty.
It doesn't matter. Yeah, I saw a little clip that Rob Skiba put up with the similarities, and they explained a little bit how they did that. So they took out the bits that were alike, and but they left over a lot of the things that wasn't alike, and that took up about that 1 or 2% maybe, but still also the sequence of the whole DNA is also very much different than all the things that are alike. And it was a really very good explanation of how they sort of did that and make it seem that it's for 99% similarity.
Yeah. They already said that they throw out data. That's reassociation connects. Yeah.
Yeah, it's amazing. And that's science, right? We throw out the junk that we don't need or don't have explanations for, and then we make a story around the rest that we have.
Exactly. Well, there is no science in there. I didn't hear a hypothesis. I still haven't heard one. In 12 years, I haven't heard a hypothesis for evolution. Not a single viable one. All right.
Yep.
We ready to roll?
Yep.
Chromosome 2 fusion. Human chromosome 2 fusion. This subject, I couched this with Professor P.Z. Myers because he decided to go on the Steve McClown of the Clan McClown's channel and start boasting and champion the human chromosome 2 fusion. And what he was attempting to do was Jeffrey Tompkins, a PhD from the Institute of Creation Research, was writing on this topic, right? And P.Z. Myers was providing all the rebuttals. And this was a big brouhaha. It happened 2 or 3 years ago. I did a complete exposition on Steve McClown's Clown Show and P.Z. Myers. P.Z. Myers got sent a copy of the show that just took this and beat it about the head and shoulders with a baseball bat. And I never heard from P.Z. Myers. I still haven't heard from him. You're not as dumb as you look, sir. I'll tell you why he never showed up. So, P.Z. Myers, this was his claim. Why are they arguing with it, they being creationists? Why go around denying the physical evidence that's out there? Oh, PZ, we're laughing at it and you. Jeffrey Tompkins, PhD from ICR, and or anyone else for that matter, shouldn't have even entertained, much less written on, the complete argument from ignorance, affirming the consequent, and begging the question fallacy, the Triple Crown. Congrats! To begin with. Because all P.Z. Myers has is synteny. What's that? It's a term used to describe the state of 2 or more genes being present on the same chromosome, though not necessarily linked. Right? That's all he's got. But what is scientific evidence? It's the testing of a hypothesis or theory that is objective in a controlled environment. So, I know this is going to sound like Greek to you, but where's the formal synteny scientific hypothesis, then experiment, i.e. hypothesis test, that validates your physical evidence, PZ? That's what we need. We're talking science here. And since there are actually 2 hypotheses for every experiment, that is, the alternative and the null, post the null hypothesis that was rejected or falsified. Audience tip, don't hold your breath, because they don't, never, and never will exist. And remember, no hypothesis equals no experiment, which equals no science. Hard stop. So all that's left, like Max Planck said, is poetry and imagination. So here's what they do. They look at genes, concrete nouns, no phenomena, skip the first step of the scientific method, make a comparison, then conjure up a story, a just so one, then ask us to disprove it. Ah! You think you got us. It's the acme of foolishness to even attempt to disprove a complete arguments from ignorance fallacy, Sherlock. Do you find it scientific or logical to imagine things, then have other people attempt to disprove your imaginings before you give scientific evidence of your imaginings? This is tantamount to claiming that invisible three-toed gnomes throwing pixie dust in a black hole behind the Crab Nebula are responsible for creating dark matter. Then you ask us to disprove it. If we can't, therefore your claim is true. Does that sound logical to you? All this nonsensical buffoonery chromosome 2 fusion is just another fiasco in the never-ending post-hoc observation parade of repackaged evolution fallacies, vestigial structures, junk DNA. We're going to get to that next week. Ring species, fossils, punctuated equilibrium, Piltdown Man, Lenski's E.coli, which is coming up, to establish universal common descent or the tree of life. That's it, right? And if you're unsure about the tree of life, if you think we haven't pummeled that already, go back to part 1. Questions, comments?
I've got a bit of a question more than anything. If you just scroll to the chromosome 2 stuff, because I get a bit of genetics, but I don't profess to, fully understand it.
What do you need, sir?
Right, so chromosome 2, right, this is again, seems to be another case of mispurporting the facts to support your narrative. So the supposition is that within apes,
right,
the 2 different types of chromosomes are found within apes that match this.
Hold on 1 second, Adam.
Well, let me get to the end of my ramble.
Okay, go ahead.
So the point of their supporting argument is that these matching chromosomes that are also found in chimpanzees, I think in gorillas and orangutans as well. But the important part about chromosome 2 is that it's a fusion of these 2 things, which is not found in any of them previously things you wanted to link it to. The only way you find those 2 different sequences is in 2 separate genomes. So it's actually a significant show that they're not related in the slightest. Whilst there may be those sequences, they're not contained in any way significantly. Like I said, they're in 2 different chromosomes. So you're talking about programming, just like having a similar piece of coding, but you've put it in a different place completely. One's got it duplicated and one's got it conjoined together. You're talking programming. That's not a support to suggest that the 2 DNAs are similar.
I wanted to interrupt you because I forgot to preface this, and I should have. I shouldn't assume that people know. Why are these guys attempting to do this? Well, humans have how many chromosomes? How many pairs? 23. Guess how many apes and chimps have? 24. That creates somewhat of a problem, right? So how do you fix the problem? Well, you just conjure chromosomes fusing, right? Sorry, I didn't preface that. I should have. I thought of it afterwards, but go on. Go ahead.
You're right. That's the trouble with having a rib removed.
Yeah, right.
So wait a minute. They had to make it fit and therefore came up with that fused chromosome?
Exactly. You got to make it fit. If your narrative states that, well, humans come from chimps or what, it doesn't matter, right? Then you look at the chromosomes and the number's off. Well, then you got a problem, right?
Huh?
So they just tailor the story. It's a shapeshifter. They just tailor their story, and that's all it is. There's no science here.
That's why they can make predictions then, huh?
That's right.
I can make predictions that way too. I mean, make up a story on long ago, right?
That's an ad hoc fallacy. I mean, and it's telling that in the state where I live, in Ohio, some years ago, they debated at the state level whether or not information contrary or challenging to evolution should be allowed to be taught to high school science students, which tells you everything about it. Because if they have to debate whether to tell high school students the whole picture, then you know this isn't about science at all. It's about controlling information so people will reach a foregone conclusion, as they do in all the public government schools. They will leave out things and tell you what the others, the other evidence is without showing it to you, if they do that at all. And then they get a predictable result. People believe it's true. And so they can only do that by not only the legal angle, but also, like you said, we're saying about the ad hoc explanations about making up fusion or vestigial organs or whatever it is. It's all a philosophy from the start.
Agreed. Similarities is not evolution. Similarities is similarities. I mean, animals have eyes, humans have eyes. Animals have legs, other animals have legs of some type. Four-legged animals, two-legged animals. These are just similarities.
True. And you have different kind of cars, but you have different sort of dealerships with cars, right? I mean,
yeah, I've never seen a barracuda give birth to a whale.
Nope.
Well, that's where the problem lies, right? You got people making statements like that. Yeah, we never seen another kind or whatever, or barracuda the whale, right? Evolutionists, they're saying these things because you haven't defined evolution yet. So don't laugh at them. They're just hearing what they pick up off the street or pick up, in hangouts and stuff. It's not their fault. You have yet to define it for them. So whenever you hear somebody say something silly like that and say, that's not evolution, well, define it, and then we'll see. But before you do that, they're fine. They're good, because that could be evolution. Why? Because you haven't defined it. No one has. See the problem? So don't be laughing. I'm going to catch you. Ain't going to be pretty.
Okay, can I do some admin, some shout-outs? If I have the voice.
Yes. Or you lose your voice.
Okay, so we have a super chat from Rakia Life, and he says, "Take my money, I'll just climb a tree and get more. Shout out to long arms and fingers that grip."
You can make money grow on trees, can't you? Or grow out of your armpits. Why haven't we evolved something like that? Right? Because everyone needs more money. That would be beneficial. But why can't I grow a couple hundreds out of my armpit? Yeah. That sounds silly, right, evolutionist? Yeah. Well, I'm saying that could be evolution because no one's defined it yet. I'm still good.
I'm waiting for these trees to run away from woodchoppers and stuff.
Woodchuckers from beavers.
Also, yeah.
You call them woodchuckers. Is that what they call beavers in Netherlands? Do you have any beavers in the Netherlands?
No.
No? Do you have any trees in the Netherlands?
Very similar.
Hold on a second. Do you have any trees in the Netherlands, first off?
Yes. Surprisingly.
Listen,
But
I'm not making
few beavers.
How many you got? How many beavers you got?
I don't dare say.
I don't dare say. I thought everything was...
all the beavers in the Netherlands are damned. Don't worry about it.
Or dikes. Sorry.
Oh my God.
Sorry, I just drifted off for a sec. I was just checking the football. What are we talking about? Betty's beaver?
No!
Hey?
Thanks, Adam. That's what it needed.
I can't take you guys anywhere.
Oh my God. I didn't mean that like that. Oh, jeez. See, and they say guys' minds are in the gutter. No.
It was on the football. I just misheard. Sorry.
Oh,
Football fantasy?
all right. Where were we?
I think you were making money grow out of armpits.
Oh, that's right. Rakia life. Go on, Moot. Or pass. Do something. And grow some money out of those armpits. All right, so human chromosome 2 fusion, that's why they do it, folks. They had to come up, because their narrative said as much, and because there's a different number of chromosomes, or pairs of chromosomes, in between us and chimps, and I think orangutans, apes, a couple other ones. You have to come up with a story to fix that. You got to plug that hole, right? Yeah, so that's what they did. Like I said, there's no science in it. It's a joke. All right, we ready to continue?
Yep.
If we're not, I'm going to anyway. So, the next fiasco. Let me get set up here. We got it. All right, Captain. There be whales here. I don't have that Scottish accent. Does everyone... Adam, do you have a Scottish accent? You could do that, right?
Right there, then.
Oh, good.
What? I'm trying not to sound like Groundsman Willie from The Simpsons, but Cartman, there be... No, that's a pirate accent. Can we go with a pirate? I can do a pirate. Cartman, there be whales here.
That doesn't sound like Scotty, man. Somebody beam you up.
Yeah, I liked it. Yeah, it's good.
Did it sound okay?
Yeah,
It was more piratey.
All right.
Yeah, it was very good. It was very good.
Yeah.
Ship ahoy. Let's go.
Chips.
Ahoy, hoy.
So here, I'll tell you what, folks. You want to see a belly laugher. This is going to be a quick one. So we got Captain Nerby Wills here. This is the lineage down to whales. And what I'm going to do is I'm going to take Pakicetus right here and Ambulocetus right here. Well, I'm not going to do anything with Ambulocetus and Rhodocetus. I'm going to let the guy that conjured this nonsense, Dr. Philip Gengridge from the University of Michigan, pummel it himself. I'll just take care of Pakicetus. So we're going to take 3 of these out to the woodshed. Okay? And this is
The
absolutely...
woodshed?
Yeah, we're going to take them to the woodshed.
Do I need to take out rolls?
All right, so like I said, I'm going to do Pakicetus first. Let's go right here. So here you go, folks. This one's going to be easy. From their alma mater wiki, right here, right? Are you ready for this? Pakicetus is an extinct genus of amphibious cetacean of the family Pakicidae, or Pakicidae, whatever, which was endemic to modern Pakistan during the Eocene. The vast majority, listen up, the vast majority of paleontologists regard it as the most basal whale. Folks, you're looking at a frigging whale. I'll shut up for a second.
No, that can't be. It's a dog. Sort of.
Listen, nonsense.
So a strange kind of dog, by the way, but yeah.
Folks, you know, Betty, I think I figured out why the top of its head's missing.
Yeah,
skull.
it's not complete, is it?
No, it's a transitionary phase. It's that hole in the head that the whale's going to get. It's just that first
Well, where's
stage.
his eyes then?
It had a soft skull, which is how it will breathe through the top of its head later on.
Oh, I'm glad you explained that to me because that's.
Or move its nose back.
Yeah.
So, folks, do you get it? Right? If I have to explain, oh my God, if somebody says that's a grown adult that's not delusional, that they think this is a whale, the vast majority of paleontologists regard it as the most basal whale. I'm sorry. The prosecution rests, right? But we'll continue. It goes on. Somewhat more complete skeletal remains were discovered in 2001, prompting the view that Pakicetus was primarily a land animal about the size of a wolf, very similar in form to the related Masonichids. Thiessen et al., he's an expert in this area, 2000 one wrote that pachycetids were terrestrial mammals, no more amphibious than a tapir. Okay? So, like I said, I really don't have to do anything here. They take care of my business for me. So, like I said, they're okay. I'm sorry. Anything?
No, I'm just looking at the phraseology from Pakistan, Eocene. I mean, they just make this stuff up. Where's the evidence for it?
From the Pachycetidae.
Yeah.
This is the evidence.
It's made to sound like it's informed, isn't it? But
yes.
You can see from the root derivatives that it's just clearly a Latinized version of saying it's a dog thing from Pakistan.
Yeah, exactly, Adam. You got it. That's their game. Yep. Boy, that sounds valid. Oh, you must be a scientist. Right? Unbelievable. All right, like I said, that takes care of Pachycetus up here. So he's gone out of the lineage, and that's at the base, so we're all over it. And besides that, what's evolution? We still don't know that. Of course, that they say this is evolution. Like I said, I'm going to let Dr. Philip Gingrich take a look at this gentleman right here. You guys got this. We're going to this is Dr. Philip Gingrich of the University of Michigan that conjured most of this mess. He's a subject matter expert. We're going to let him tell you about Ambulocetus first and then Rhodocetus after that. And then I'll check back. We'll go over some of his comments. So listen up.
Maybe slight sideline. I think that mostly because it's unusually large. The eye.
So Ambulocetus is not on the main line.
Oh, for sure. Bispect.
There you go, folks. Like I said, he does my work for me, right? Professor and director.
Embarrassing.
Hold on a second. Professor and director the Museum of Paleontology at the University of Michigan, Ann Arbor, the subject matter expert that conjured a large portion of this mess. You heard what he said, right? Let's go with Ambulocetus first. Has its eyes raised up on its head in a very strange way and is unusually large for an early whale. Maybe it's not on the main line. You think? Can you please list the bones that were found for Ambulocetus? This top A is a reconstruction of Ambulocetus at the end of the power stroke during swimming, like I said, by Thweson. He's one of the experts in this field. The actual bones that are found for Ambulocetus is B. The stippled bones were all that were found. The bones colored in red were 5 meters above the rest. Let me tell you something, folks. I had more bones in my chicken last night. So it's not on the main line. It's not on the line. It's not on any line. This is all conjured. And this is going to tell you right now. Erotocetus, these guys, he even told you, more or less, that they painted flippers on the front and a fluke tail. They didn't have any of this and still conjured this mess. They not only conjured it, they showed pictures of it or made diagrams of it. To how many thousands of kids and people that walk through that museum? I'll tell you what, I'll bet you it's still the same way today. I'd like to see somebody go to that museum and go through it and show us Rhodocetus. Dr. Phil, I speculated that it might have a fluke tail. I now doubt that Rhodocetus would have had a fluke tail. Translation, Dr. Phil didn't have the tail, so he conjured it. Then he drew it in. That's science. Dr. Phil, since then we have found the forelimbs, hands, the front arms of Rhodocetus, and we understand that it doesn't have the kind of arms that can be spread out like flippers on a whale. If you don't have flippers, I don't think you can have a fluke tail and nearly powered swimming, right? This is a flippin' joke, folks. And like I said, I don't have to do any business here. They've done my work for me. Questions, comments?
Yeah, the narrator's voice, it was whispering seriously. Scientist Gingrich has to fill in the gaps like fins and flippers. I mean, what the heck? You find one or two bones and you think you have something.
Adam.
Amazing.
I think, can you just scroll up to just the end bit where the summary of his statements with regards to, just that's it there, look. Since then we have found the forelimbs and the front arms of, that whole statement for me says it all. It confirms that whatever his learned opinion was beforehand, it was based on absolutely no knowledge of what those limbs were. So the only supposition they used to build the model of this animal was the paradigm they already had. It wasn't based on the fossil itself, because they didn't have those bits. So they made the bits up around it. And surprise, surprise, what they made up was totally wrong. And the trouble is that, and this is endemic in the whole of this field, is that these things linger. They still hold up the narrative, even though they're disproven. Lucy the Australopithecine kind of nonsense. They're all there. They're still cited, and it's even when disproven, and they've been the fundamentals of. I mean, this I assume at some stage was a fantastic example of an evolutionary chain. But,
I'll bet you it still is.
But as more and more actual evidence comes to light, it shows a complete non-linkage, as displayed by the position of the eyes in the head of these animals, and that all they're doing is just pitifully linking these things. And the only validation, and it's not real validation, appears to be the stature of the person that builds the story.
You got it, Adam. Paula.
Yeah, that's exactly it. It's about the status of the person making the claims. If they have the degree from the university and the peer review, which again is an echo chamber, but if they have the right credentials that pushes the narrative, then they are believed blindly, even when they contradict themselves, like you were just showing, how they say one thing and then say, well, I kind of made that up. It wasn't true at all. And they call this scientific progress, as though they really thought it was had the flippers and some such before, and then discovered, no, well, maybe that's not the case. That's not what happened at all. They made it up and then found a reason to abandon that story and go with a new one. I mean, and this is common. Like Lucy, for example, that was supposedly this first humanoid whatever. if you look at the actual bones found, there was just 5 or 6. There were just a paltry amount of bones that were spread over a wide area. And this is common whether it's distance or depth. These bones are typically not found together. And if they are, they often find, well, this was actually not, like the one where they thought that there was all these little dinosaurs, or they were just juveniles. The story keeps changing, not because science keeps improving, but because they can't keep their story straight for long enough, and so they have to keep inventing new ones.
Yep. Betty.
Did you call me?
Yeah.
Okay. Yeah. Well, everything has been said. I just someone in the chat that says to me, right? The guy named Supercurve says, "Yeah,
God.
Betty. So where are Eviscerated today? Answer me that. Were they designed and then made extinct straight away? Lazy." So, because we have
sorry, Betty, was the question, where are those dog-like looking animals in Pakistan today?
Yeah. I think so.
Pakistan?
That was an awkward silence. We knew where you were going to go. Well, ain't they in Pakistan?
Yeah, that's for dramatic effect, I can understand. Yeah.
I don't get the point of his quote. What's your point?
Well, the thing is that you have these strange skeletons that I find, the T-Rex, all other kinds of skeletons, and then we don't see those skeletons nowadays anymore. When our dogs die or whatever dies, right? So it's something that we don't know, or how it looked like. And since it's now dead and it isn't there anymore, it must have evolved into something else, according to the guy.
I don't know. I don't know. Did I state an argument where things don't go extinct at some point here? Can somebody remind me?
John, if I was making shit up, right, and I had that skeleton and I was going to try and claim evolution, I'd try and claim it evolved into like the North Pakistani mountain wolf,
right?
Not a freaking whale.
Very good, Adam.
Yeah.
We
did have a big flood, of course, so I think that most of the animals were dying in a big flood.
You think?
Yeah, I do think. Therefore, I am.
There's a book comes to mind. It's called Bones of Contention by Marvin Lubenow. He goes through all of these things and just tears it apart.
Yeah, I heard of that book. I haven't read that book. I think did Paula remind me of that? Paula, did you read that book? Stop washing clothes, Paula. Did you read that book? Somebody recommended that. I thought it was Paula. I've heard of that name before, and I haven't read through it, but yeah. All right, we ready to continue?
Yep.
I reckon so. I don't know about you, Temp, but I had a whale of a time on that topic.
Well, it's very current to the discussion.
Listen, oh my God. Yeah, but you didn't hear him because your ears were filled with blubber.
Hang on, let me get the scales out of my eyes and we can go on.
All right, folks. That was just the preamble to this. Lenski's E.coli. The LTEE. Right? What a joke. This belly laugher holds a special place in my utter contempt amateur night belly laugher file for many, many reasons. For the first year and a half or so on the Evo retard circuit, I never heard this claim. For being a top evolution proof, in retrospect, it raises a serious eyebrow, doesn't it? You'll see why that is over the next 10 minutes. Like I said, I never heard of it prior to an Evo Clown parroter bringing it up. Of course, it was the usual superficial appeal. What about Lenski's E.coli? I was like, well, what about it? In this particular case, I wasn't sandbagging him. It wasn't a setup at all. I actually knew nothing about it. Right? I can vividly remember asking one of my fellow Evo crushers on the forum what it was, and he said that the retards think E. coli mutated and developed the ability to metabolize citrate. I was utterly gobsmacked. Not at the claim, but at the utter gullible, moronic stupidity of these mindless parroters, Dunning-Kruger retards. Listen one tap. I'm going to get real close here. Listen. A first-year biology major would belly roll in elephant tears at this buffoonery. You don't have to be an expert with E. coli. All you need to know is it's a facultative anaerobe, i.e. it can survive in oxygen and oxic environments. I'll expand on this momentarily. Right? Now, so we don't strawman anything, I pulled this up off of their alma mater wiki. Let's go through it. The E. coli long-term evolution experiment, LTEE, is an ongoing study in experimental evolution. Really? What's evolution? Led by Richard Lenski, and there is no experiments, that has been tracking genetic changes in 12 initially identical populations of asexual E. coli. Really? Do you have sexual E. coli as opposed to that? My word. Since 24 February 1988, the populations reached the milestone of 50,000 generations in February 2010 and 66,000 in November 2016. Lenski performed the 10,000th transfer of the on March 13, 2017. Over the course of the experiment, it's not an experiment, Lenski and his colleagues have reported a wide array of phenotypic and genotypic changes in the evolving, whatever that is, populations. These have included changes that have occurred in all 12 populations and others that have only appeared in one or a few populations. For example, all 12 populations showed a similar pattern of rapid improvement in fitness and decelerated over time, faster growth rates, and increased cell size. Half of the populations have evolved, whatever that is, defects in DNA repair that have caused mutator phenotypes marked by elevated mutation rates. The most striking adaptation reported so far is the evolution of aerobic growth on citrate, which is unusual in E. coli, in one population at some point between generations 31,000 and 31.5. Everyone got that? People, it's painfully obvious to anyone past the first day of studying metabolic pathways that E. coli, right? Remember, it's a facultative anaerobe. E. coli already had the metabolic machinery to deal with both substrates, glucose and citrate, right? It screams simple operon, then promoter and repressor issues, as in a citrate transporter gene being switched on whether oxygen is present or not. That's all it means. That's all this. Even before I ever looked into the topic, I knew that it had to be this issue right here because it could already metabolize citrate. I already knew that because of aerobic metabolism. We're going to get into that here because it's called the citric acid cycle, right? Citric acid, citrate, you get the connection? Citrate's just a derivative of citric acid. So, this is their claim. E. coli developed the ability to metabolize citrate, whereas it supposedly could not do so before. And that equals evolution, eh? Are you kidding me? Crayon munchers. E. coli already possessed the ability to generate and utilize citrate in its normal oxidative metabolism of glucose and other carbohydrates. And E. coli is normally capable of utilizing citrate as an energy source under anaerobic conditions. Dunning-Kruger knuckle-dragging numpties who think that metabolism is the religion of self-referential cereal bowls postulate again, E. coli developed the ability to metabolize citrate, whereas it supposedly could not do so before. Yeah. However, this is clearly not the case, like I said, because the citric acid cycle, the tricarboxylic acids, or TCA cycle, or Krebs cycle, all names for the same metabolic pathway. You see this citric acid cycle? Citrate, citric acid, do you get it? I know you don't. This is Greek to you. I know it is. All names for the same metabolic pathway generates and utilizes citrate in its normal oxidative metabolism of glucose and other carbohydrates. Get ready. This is the death knell. From Poss, Dimroth, and Bott, the E. coli citrate carrier CITT, a member of the novel eubacterial transporter family related to the 2-oxoglutarate malate translocator from spinach chloroplast, the Journal of Bacteriology. Listen to this closely, folks. E. coli is normally capable of utilizing citrate, as an energy source under anaerobic conditions with a whole suite of genes involved in its fermentation. This is the key point. This includes a citrate transporter gene that codes for a transporter protein embedded in the cell wall that takes citrate into the cell. Friggin' boom. That's it, right? Listen, folks, that's all this boils down to. Let's say you're living at home and there's a bread blockade. There's no flour in your neighborhood. Are you going to be eating bread? No. Let's say a bread truck finally breaks through and busts into your living room and explodes and 50,000 loaves of bread fly all over your house. You're probably going to be a bread-eating sumbitch over the next 5 months, aren't you? That's what this boils down to. So again, E. coli evolved and natural selection selected the ability it already had in the first place, eh? That's amazing. So Lenski, Dick, went looking in the DNA for what changes occurred to allow E. coli to metabolize citrate in an oxygen-rich environment. And what did he find? All that happened was an already existing oxygen-tolerant promoter. We're going to go over this stuff here in a minute. Give me about another 2 or 3 minutes. An oxygen-tolerant promoter had been moved next to the CITT gene, which allowed E. coli to then metabolize citrate without oxygen present. There wasn't a new functioning element created here. It was already in the genome, right? It just wasn't being expressed in an oxygen-rich environment. There's nothing new here. Nothing new was created at all. Let's see. From Blunt, Barrick, Davidson, and Lenski, genomic analysis of a key innovation in the experimental E. coli population of nature. It is important to note that E. coli, here we go again, already have the ability to transport and metabolize citrate. At least they're honest. But the bacteria typically cannot do so in oxic conditions, as it does not produce an appropriate what? Transporter in this type of environment, among other required factors. No, that's the main factor. The genetic changes that underlie this particular adaptation are complex, but a key involved the replication of a genomic region that regulates a what? Citrate transporter. This amplification captured a previously existing anaerobically expressed promoter, the promoter for RNK, which could then direct transcription of the citrate transporter, CITT. Repeated tandem amplifications refined this function. Like I said in the beginning, before I even got here, I said promoter, operon, repressor. It screams it. Again, Stanek, Cooper, and Lenski An identification and dynamics of beneficial mutation and long-term evolution experiment E. coli evolutionary biology. The researchers found a small insertion mutation upstream from GLIM-US, an operon involved in cell wall biosynthesis. It was hypothesized that this mutation inhibited normal binding of a transcriptional activator to this region, thereby reducing GLIM-US expression. And again, Schneider, Duberke, and Lenski, Dr. Lenski and colleagues observed an insertion mutation into the NADR gene and hypothesized that an increased intracellular concentration of NAD may be beneficial in this environment. This increase in NAD would be due to a loss of function in the repressor component of the protein. Like I said from the beginning. I mean, anyone, I'm telling you, first year biochem student laughing their butt off at this. I'm just saying. So let's just take an overview of this. This isn't going to be a detailed instruction in operon promoter repressor functions, okay? This is a complete operon. It's a suite of genes. In this particular case, I snagged the tryptophan. It's not that important. It's not the same as what we're talking about. However, the concept is the same, right? So you got a promoter on this gene, and what it does is, it's like the RNA polymerase. It's like, hey, big guy, what you doing, right? So it promotes RNA polymerase, the copier and tape reader that's gonna copy this and send messenger RNA to go ahead and express these genes, right? So you got the promoter right here, and RNA polymerase attaches a little bit upstream from the operator. However, in this particular case, you have a repressor, right? For tryptophan. So how this works is, it's an intense feedback loop. If you got a lot of tryptophan in the cell, it's a feedback loop. Tryptophan attaches to the repressor. The repressor has a conformational change and attaches to the operator. This is a break or a stop sign for RNA polymerase. Makes sense, right? You don't need to make tryptophan if you already have enough tryptophan in the cell. No intelligent design here, folks, right? I digress. But right, it's as simple as that. That's all it is, right? And like I said, down here, you can see it. The whole operon, the repressor's knocked out of the way, and RNA polymerase can just go ahead and start, transcribing this section of genes, right? Just like an E. coli. Promoter, operator, repressor issues. That's it. In conclusion, after 25 years and 66,000 generations, all Dick ever gets is E. coli. E. coli could always metabolize citrate. The only reason it couldn't metabolize it in an oxygen-rich environment is because it was switched off while oxygen was present. That's it. All that happened was a promoter was moved next to the switch that basically kept the citrate metabolizing process, the citrate transporter, CITT, stuck in the on position. It always had it, it's in its genome. That is, a regulatory mechanism broke. That's all that happened here. Metabolizing citrate in an oxygen-rich atmosphere may be a new thing for E. coli, but being able to metabolize citrate in the first place wasn't a new ability. Breaking an on-off switch within a genome may have had a beneficial result, but it's not a new creation of anything, like I said above. As a matter of fact, Dick is a creationist's best friend, showing nothing more than an already existing ability being broken. Bottom line, this is their claim: E. coli's aerobic growth on citrate equals evolution. And again, the only reason that is the case is that in the presence of oxygen, the production of the citrate transporter protein that is inside the operon was switched off. That's it, folks. Now I'm going to take the nail spike Louisville Slugger to its head. Are you ready? This is going to go quick. This entire fiasco is tantamount to saying, while attaining a $100,000 annual income, we ate ribeyes. But since I was laid off and now earn $19,000, all we eat is Spam. We're evolving. Listen closely to this. call Dick and have the culture medium switched from glucose and citrate to glucose and silver, glucose and cyanide, glucose and hydrochloric acid, glucose and saccharin, or glucose and silicon. Then whittle away the glucose and let's see what happens, Dick. My word, crayon munchers. Also, at any point, did E. coli stop being E. coli and a giraffe or a duck-billed plesiosaur appear in the dish? My word. And may I say again, from Pierre Grasset, Evolution of Living Organisms, back in the day, he told you back then, bacteria, which are both the first and most simple living beings to have appeared, yeah, like you knew that, are excellent subject material for genetic and biochemical study, but they are of little evolutionary value. Whatever evolutionary means, we don't know that yet. Right? So this is a joke, folks. Right? Last shot. From Wikipedia in the E. coli spiel. Listen to this closely. Some bacteria, notably E. coli, can produce and consume citrate internally as part of their TCA cycle. Why did they call it this TCA cycle? Why don't just call it the citric acid cycle? That's what the article's titled. Why switch the name? Yeah, right. As part of, now they're correct, but why didn't you just call it the citric acid cycle? Why? Because it may impugn some citrate issues? Unbelievable. Let me go over it. Can produce and consume citrate internally as part of the TCA cycle, but are unable to use it for food. Listen to this. Because they lack the enzymes required to import it into the cell. What? Number one. So E. coli can produce and consume citrate, but are unable to use it for food? What in the world? I suppose they think their erroneous appeal to internally covers them, eh? Get ready. I'm gonna friggin' jack this yard. Number two, enzymes don't import substrates into cells. You clowns, transporter proteins do. Where the heck did you come up with it? You're just making this up. Boy, that sounds sciencey. Let's just stick with that, huh? No one's going to catch that. I'm going to tell you why here in a minute. So this statement is nonsensical, right on its face. Turn out the lights. Listen to it. Remember, they lack them. I'll just put machinery because this is a joke. They lack the machinery required to import it into the cell. What did we just go over above? About the CIT transporter, right? Let me show you something. Because turn out the lights. The party's over. Again, from Dimroth et al. E. coli citrate carrier, CITT, a member of the novel eubacterial transporter family related to the 2-oxoglutarate malate translocator from spinach chloroplast, Journal of Bacteriology. Now, remember what they said. They lack the machinery required to import it into the cell. E. coli doesn't have the machinery, right? Really? E. coli is normally capable of utilizing citrate as an energy source under anaerobic conditions with a whole suite of genes involved in its fermentation. This includes a citrate transporter gene that codes for a transporter protein embedded in the cell wall that takes citrate into the cell. You're saying it doesn't have this machinery required to import it into the cell. What does this say? What on are you just making stuff up? Listen, this is the game, folks. This is how the pseudoscience high priest God deniers roll. It's a numbers game. Just humor me. They know they can get away with this utter belly-laughing bollocks because the vast majority, that is 99.99999% of the population, wouldn't know the difference between metabolism and a chartreuse windsock. Let's say 0.001% of the population understands some aspects of metabolism. Within that minuscule number, you're going to have one, EVOs. For the life of me, I can't see why. I can't see why you're an Evo with this and whatever that is. Number 2, people who aren't paying attention, which is most people anyway. And number 3, people who could care less. That's going to take the paltry 0.001% number and puts it into the 0.000001% area. That's a pretty tiny number in the scheme of things, don't you think? And can be easily drowned out, i.e. beyond barely palpable background noise. So they'll just take the hit for that. It's not a hit, right? Because no one can hear them. No one's going to hear me, right? Do you see it? Unbelievable. Questions, comments?
Yeah, I just want it because it's a big picture sort of thing that I'm more prone to because I'm not deep into this like you are. But I think overall the complexity of biology that they have been discovering was unanticipated by the theory of evolution so bad that they have to get into these little tiny details that people don't understand, like you were saying, in order to sell it on some level that most people are happy with, like you said. And they'll parrot things like antibiotic resistance, like you were mentioning earlier, that the bacteria already had the capabilities, but when you put out an antibiotic that kills off the ones that don't, all you have left are the ones that do, and now they don't have any competition. So the same bacterium that you had before has now become resistant because you got rid of all the competition. And that's what they think is bacteria changing from something to another thing, when all it's doing is just people ignorant or deliberately hiding the fact that they already had the ability. And that selection is always doing that. It's always taking a subset of what already exists, getting rid of the rest that it doesn't select, and reducing the gene pool as it goes. And so if your antibiotic depended on a certain capability that doesn't exist anymore or has been suppressed, then you're doing the opposite of evolution. That natural selection is the enemy of evolution because it keeps reducing the gene pool and making things less and less capable. And it's just a giant pile of contradictions and denial of what they themselves say from one day to the next in biology or whatever discipline it might be, to use lack of a better term than discipline. But anyway, that's just my thoughts on it. Sorry.
10th.
I'm still hung up with how they figured out the bacteria was asexual.
Did you like that? I thought I read that. I started laughing the first time I read it. Right?
Maybe some identify with women. Get it right.
I'm trying to say that's a greater feat than anything, to figure out which bacteria wants it and which one doesn't. I mean, that's pretty big.
What kind of bacteria?
That's the craziest thing I ever heard. This bacteria
based on
has no... Go ahead, Adam.
I was going to say, based on the colour of the flagellum, I think, but...
that was weak.
Pink or blue? What?
Yeah, no.
So they're gonna do a long-term evolution experiment with bacteria that they say is asexual, and then they come up with all these terms that 99.99%, and I'm one of them, wants to just turn off this so-called experiment because it's just ridiculous. What does it prove?
Nothing. So it already had the ability to metabolize citrate. As soon as the minute I heard that, I said, what on earth? That's why I said, in retrospect, that's why I never heard this proof. In the first year and a half, I got bombarded by everything but it. It's so funny. I finished this presentation. I was finishing up the Lenski part of the presentation Thursday evening, right? And I said, I'll take a break. I'll go into Discord. And I'm sitting in Discord. The first person says something about evolving. So I said, evolution, what's that? And guess what they come back with? Lenski's E.coli. I couldn't believe it, right? I started laughing. I said, 2.5 years, I never heard this. I mean, it's been longer than that. Right? And then the same day I get done with it, just a few minutes before, these guys, and they all parrot it, right? Lenski's E.coli, right? I say, okay, what's that? Lenski's E.coli. See, they can't do anything, right? They got no depth. Even if they did, they have no idea what metabolism is, the Krebs cycle, right? The TCA cycle. Do you like how they switched up the citric acid cycle, right? They don't use that phrase anymore. Adam, did you catch that?
Yeah, it would be much too descriptive and not sciencey to imply it's just the citric acid cycle. I've only got 2 bits really, John. The first, if you scroll to the start.
Oh, sorry about that. The people on the outside are nuts. Hold
on
a second, Adam. I gotta let everyone. You're the only one seeing the presentation, or anyone in the Hangout. It's not going out to the audience. Give me 1 second.
Okay, it's not a major point. The only point really was really your pickups as you were reading it with regards to the word experiment. But when you do get there, if you do see it, they do qualify it themselves.
Yeah, where do you want me to go?
Just to the top with regards to the. Keep going. Did you start? There you go. Okay, so with regard the E.coli long-term evolution experiment is an ongoing study. It's not an experiment; it's an ongoing study. So I'll give them the. I don't know why they feel they need to use the word experiment when it's an ongoing study. That was just a minor point. The only thing for me again was to reiterate the point that they're misrepresenting the facts. This isn't anything again. This was already there, and if we, as you've done, analyze what's going off, what we actually see is that this thing was already had the ability to do that, but for some reason it had been switched off. Now, that is not, from a genetics point of view, an example of evolution in any sense. Again, it's quite the contrary. It's an example of preemptive programming. It's a backup system that's already embedded in the organism, just in case you need to have this ability to maximize citric acid metabolism. Here, have it, but we'll switch it off for everyday use. Now that's indicative of, frankly, intelligent designing.
Yeah, duh. Yeah, it's a load of nonsense. As soon as they said citrate with E. coli, I started belly laughing, right? Because I know the citric acid cycle. This is so stupid. It's hard to put it into language why they would even bring it up, right? Like I said, all they did was whittle away the glucose, and yeah, it started metabolizing citrate. Wow, that's amazing. Well, put that with hydrochloric acid. I want glucose and hydrochloric acid, and start whittling away the glucose. Let's see if E. coli can adapt the ability to overcome hydrochloric acid. Right? Or saccharin. Or any of the other ones I posted. This is a joke, people.
So the part you said it has a built-in on and off switch for regulation, they missed that?
No, they know it. They like I said, I had the citations right there. They admitted it, right? The operon, the repressor, and the promoter issues, right? They admit it, but no one seems to catch on. That, like I said, the general population not going to catch on. They'll just put evolution in front of it by fiat. Well, this must be evolution, right? Because none of these people, or none of the general population, or very little, like I said, know what metabolism is, know the Krebs cycle, or I'm just going to start, I used to call it the Krebs cycle for many, many years. I'm switching citric acid cycle, right? Because citrate is a derivative of citric acid. Oh my goodness gracious, people. It's the citrate is a intermediate.
Vitamin C.
That's why they call it the citric acid cycle, because it's that important. It's the primary intermediate. Hold on a second. You know what? Let's pull this up. Let me get over here. Just to show it. We won't get any in depth. Let's just try it. Oh, what do we got? Images. Let's get a clear one. What's this say? Oh, it ain't a big one. What's this say? This is the Krebs cycle. Oh, they even titled it the citric acid cycle. I wonder why. What is this? Citrate. The prosecution for, I don't even know, there doesn't even have to be a prosecution. Just the circus tent. Unbelievable, people. That's all I got to say. Oh, we got this guy, Mr. Whale. Sorry about that. Oh boy. Where are we at? Adam, do you finish up with this nonsense?
Yeah, I'm done. It was Benny and all, I think. I'm back to you with Lewis. But yeah, I mean, for me, it's, let's say, another example of selective choice of the facts, certainly, particularly in that case where it really, as Temp was saying, they don't mention that. To mention that would undo the whole claim, wouldn't it, that it's evolutionary? Right,
but like I said, Adam, who's even if you did, let's say you did learn about metabolism, it was 15 years ago, you gonna remember that? No. If you don't use this stuff on a daily basis, it's hot. It's what they call a highly perishable skill. You'll forget this stuff, man. Right? And they bank on that. Like I said, they bank on that not too many people are going to know. And the ones that do know, they're going to fall in those 3 categories I put them in. But they don't really care. There's no threat. None. They could just say this stuff and with little worry. So that really pisses me off, by the way. But it's tactically sound. I have to give them that.
Yeah, but they're holding back information that would deny the premise, so they're lying.
Well, I don't want to say that there's some overt lies, but for the most part, they don't have to lie. They can just put it out there in ambiguity and then just slap evolution on top of it, stamp it with evolution. There you go. That's it. That's all we got. So, yep. Betty, any comments?
No, I think things have been said. And yeah, it's amazing to me how they sort of they put it out there in sort of clear for everybody to investigate. And if you go deeper, you will find out that most of people don't tend to go that deep, and they will take it and parrot it. I think, not knowing what they're saying, basically. And once it's out there and repeated so much of the time, then nobody questions it, ever. It's funny.
Yep, exactly. Yep. Hey, any admin?
No, not at the moment.
Why?
Well, you got some really good admin, so I don't think that you...
That's fine.
Worry about that.
I'm only kidding. Any more final thoughts on Lenski's E.coli? So you Evos out there, you get the gist, right? Because I told those couple people in Discord, make sure and watch this week. You gonna pull out that Lenski's E.coli again to me? I don't think so. It's the same thing like what happened with spectroscopy. I haven't heard a peep anywhere about the sun and what it's made of ever since that spectroscopy presentation went off. And this is what I'm hoping is going to happen with Lenski's E.coli, right? Because you don't want to bring it out to me, because I'll jack that thing yard. I'll pull Babe Ruth and call my shot, right? Because you're not prepared for what's coming at you. You just got a little taste of it right there. All right, let's continue. It's our last section, Nylonase.
Okay.
There you go,
Nathan's
Nathan.
favorite?
Say again?
Nathan's favorite?
Yeah, I was just going to say that. There you go, Nathan. Sure you don't want to come in? Yeah, I understand, man. You sounded a little bad this morning. All right, this is pretty funny. Check this out, folks. Nylonase, this is the poster child for things breaking. I call it Lenski's E.coli redux. Same concept, similar concept going on here. Listen closely. Guess what the inventor of nylon, Wallace Carruthers, modeled his new invention after? Protein. Nylon 6 is a synthetic polymer consisting of more than 100 units of 6, listen to this, aminohexanoic acid. What's that sound like to you? Note the amino and the acid part, right? That's the substrates or the constituents of what? Protein. The enzyme that breaks down linear substrates, in this case protein in these bacteria, is the E2 and is part of the very same suite of enzymes that break down nylon. You getting it? You feel it coming yet? It's going to come like a freight train. It appears a mutation affected the enzyme's catalytic cleft. That is the active site. I'm going to go over this here in a second. In such a way that it altered its specificity, whereby allowing it to hydrolyze a wider range of oligomers, which include nylon oligomers, without compromising its original ability. All right, let's take a look at this. This isn't going to be an in-depth tutorial on enzymes. It doesn't need to be. Let's just get the overall feel for this, right? So enzymes have a specific region where the substrate binds and the reaction happens. This is called, like I said, the active site, right where this red arrow is in hexokinase. This is a yeast enzyme, right? This right here, this V is the active site or catalytic cleft. The active site is usually a cleft, like I said, or pocket at the surface of the enzyme. Enzymes are substrate specific, although specificity varies from enzyme to enzyme. Some are high specificity. The enzyme will only bind with a single type of substrate. And then you have some that are low specificity. That is, the enzyme will bind a range of related substrates. For example, lipases hydrolyze any fatty acid chain, right? It could be monounsaturated chain, it could be polyunsaturated chain, or completely saturated chain. It doesn't matter. It's that lipase is gonna take care of them all. It just has low specificity. So what had happened here in this flavobacterium is there was a mutation that affected the catalytic cleft in such a way that it opened this up. That's all it did. It's just opened it up and said, come on in. I'm open for business. They lowered its specificity. That's all that happened. And nylon being a protein, well, this enzyme catalyzes protein, right? Duh. I don't need to go on with the story anymore, do I? This is exactly like bashing in a keyhole of a lock so more keys can fit into it, right? How many new locks will you get from bashing pre-existing keyholes? Not many. You just got a busted lock. Although it still works with that original key. But now it works with a whole mess of other keys, right? Because it's opened up. It's merely a degradation of an existing ability. Nothing new. Just like I said, Lenski's E. coli redux. And again, bacteria from Pierre Grasset. We're still talking about bacteria here, right? Bacteria, which are both the first and the most simple living beings to have appeared, are excellent subject material for genetic and biochemical study, but they are of little evolutionary value, evolutionary, whatever that is. Comments, questions. This is the last section.
I think for me, John, just another good example, a slightly different form. But again, the underlying principle is it was already there. It's not been invented. There wasn't a new set of protein structuring created. It was a modification of slight, in this case, you kind of say kind of crowbarring open that molecule so that it's enabled to interact with a much wider variety of, but again, of a similar subtype. It's a little bit crude, the analogy, to use just like you know you're crowbarring it open to fit more keys in. Still very limited the type of keys that would fit in the lock.
Sure.
To work, so even though you've opened it up more, so it's just, again, it's just variation on what's there. It's not anything new.
Exactly. Yeah, this was an easy one. I knew this one a long time ago. I started snickering. As soon as I looked up Wallace Carruthers and what he patterned the enzyme or what he patterned the enzyme after was protein, I started laughing. I already knew it. I already knew where it was going, right? This stuff's, pretty simple once you get the story. But what happens is you just get people parroting, well, Nylonase. They won't go any deeper, right? When's the last time you heard an evolutionist talk in any beyond surface level of anything with Nylonase? Ever? Yeah, crickets.
Now, as soon as people hear something like nylon, it's artificial. It couldn't have been designed because this is, there was no such thing as nylon, before, when these things were made, as if the things that are designed can't adapt per their design. I mean, it's not a proof of evolution at all, but they think it somehow is, and they just shut down as soon as they make that claim. They just shut down.
I like them jack them up with this one because I do. Because the enzyme, the original enzyme, what does it metabolize? Well, it metabolizes friggin' protein. And what's nylon? Well, it's friggin' protein. It's made from amino acids. Yeah, that active site is going to be pretty similar, especially the more drastic the catalytic cleft is affected. It's a no-brainer. It's over.
I mean, John, you can kind of underplay it in a way there with that again, because this isn't like all bacteria you stick on there and they start doing that. It's just a specific one. In part of its way of standard, there's a kind of a coincidence that this then occurs, and that probability is low, which you in terms of that occurring. But again, for me, it's these kind of things that these biological systems have the ability to adapt based on environment and changing DNA coding in such a way as to benefit themselves. That's not evolution. That's responding to your environment based on pre-programming. And again, if you could define evolution, it again, for me, undermines this random mutatory effect, which is supposed the cause of it.
Hey, did you say mutatory?
Yeah. Is that real? Mutatory effect? Yeah, that's all right.
It's a mutatory from bullshittery.
Yeah.
Too funny. Oh, and by the way, if I'm not mistaken, Adam, this was a mutation also in the plasmid, so it can be passed. That mutation can be passed to other bacteria. I haven't chased that down, to be honest. I'd have to go chase it down. I don't really care because I could just stop with, right here. But it's an interesting thing if you wanted to chase that down.
Well, passing of information like that is just, again, evolve that. Go on.
Yeah. You're
I sent you a message. How did you know, Mr. Bacteria, how to read my other message? You know what I mean?
Oh, that's a bigger fish to fry. In fact, we went over that today, information in Nathan's second show that came up, Adam. So you're right on point, right?
How did the first ever bacteria know how to read what the first ever bacteria wrote?
Yeah. It's much worse than that. Where'd you get the bacteria? Where'd you get the cell wall? Where'd you get DNA? Where'd you get the lipids? Right? Not just fatty acids. You got phospholipids. Where'd you get the phospholipids from? Right? Those reactions are uphill. Big time. That's just the tip of the iceberg. We can go down that. I don't think we need to go down that trail right now. 10th. Or I'm sorry, Adam. Were you done? Anything else?
No, I'm done, mate. Thanks.
Did Tony come back?
He's not here now.
Yeah, for a short while, but I think that he left already.
Okay. His internet went bad when the football game started, the third quarter started?
Oh, that game's over.
We only have 2 halves, and they're done for him, so it must still be internet.
All right, 10th. Any thoughts on Mr. Nylonase?
Not really, other than the fact that you can take what's already there that I believe the creator made, and you can isolate it and see how he made it through whatever knowledge we've arrived to. And then, when I remember when they were cloning sheep, and it's like, yeah, it took an outside source to do it. Well, the outside source here is God, who made everything, and we find out there's on and off switches, and we find out there's adaptability built in. But where'd you get it from the beginning? And as far as I've learned, a mutation is a loss of information, and adaptability is already pre-programmed. You're going to call something adapting a mutation.
Well, you got to have something to mutate first. Where'd they get the DNA to mutate, right?
Precisely.
Right, you can have conditional...
And how did it know what to mutate to go to something else that it could need?
Right.
Well, that's Adam's point. It's got to communicate. How is it communicating with the first one? If it's the first one, before the second one gets there, how does it communicate when the second one's not there? It had to all be there from the beginning, precisely, and working to a design.
Yeah, we talked about that last week with irreducible complexity, exactly, with interlinked systems. Yeah, exactly. You got it. Betty, thoughts on this?
Yeah, well, they did a good number on us with the evolution Fairytale, didn't they? How many people are buying into this now? I mean, so many people at schools are being taught this. And I remember that I was drawing chimpanzees and then in a sort of a line, right? And then they say that we were hunter-gatherers and stuff like that. I mean, it's so ingrained into people's ideas that we descended, or our forefathers were monkeys. And I thought, well, I never bought that. By the way, it sounded so stupid and foolish for me. But yeah, you had to learn it, right? So you sat there and you thought, well, these people should know. But I remember that they weren't like feeding it to us as complete truth, but that they were still sort of finding more truth in this and try to science it. But still, I think it's now on these at these schools more that it's now established science, I think. And
Yeah.
that's wrong if you look at this.
It's peer pressure.
Strange.
Yeah, I mean,
Yeah.
if you want to pass your course, you will believe, or at least say you believe what the teacher says and the book says, and you don't want your peers to laugh at you, which, is a pretty hard thing to overcome, especially when you're young. And so you go along, and then you get out of school, and it's in the media, it's in everything, the same belief system, and nobody wants to go against that. And like like I said earlier, people have short memories. They're not going to remember enough details, but they will remember the conclusion and the laughter and mockery of people who disagreed with whatever the party line was, and people just don't want to go against that.
Yeah,
I've shared.
and it's the same as the flat Earth, isn't it? I mean, laughing at people thinking that the Earth is flat and because they will fall over the edge. But they never wondered why would someone stick on the opposite of the two, right? I thought that when it was explained to me that we were living on a ball, and I thought, well, why is this? So yeah, they repeat it, and it's indeed it's peer pressure. You have to do this because otherwise you won't. And if you get them when they're young, then and you get them also with the nice dinosaurs and these movies about dinosaurs. I mean, every kid has these toys of dinosaurs, and they love that. And you can't take their dinosaurs and their spaceships away from them. Huh?
That's right. That's
it's
indoctrination.
religion.
Yep.
Yeah. It's sad.
Agreed. Listen,
We need to wake up.
Oh, yeah. All it takes is for Micah deep study in this area. You don't have to be a friggin' expert biochemist or geneticist for this stuff, folks. I'm telling you, you don't. Right? Just underneath the surface, it all falls apart. Now, I did request somebody, if anyone's near Ann Arbor, Michigan, if they could, listen, I don't want you to run out right now, but when you get a chance over the next month or two, can somebody go to the Paleontology Museum there in Ann Arbor and go to the whale section and see if they still have Rhodocetus with flippers? I want somebody to do that. Can somebody do that out there? That would be very interesting. And a picture would help, right? And just send it, send it to Nathan, or where could they send it? You could catch me on Discord. Go ahead.
Yeah, on Discord, of course. Yeah, I like to see that too.
Well, maybe Captain Ahab, a.k.a. Phil, could send us that picture of Pakicetus and all the way to Rhodocetus.
Yeah, I'll guarantee. I'm not going to say I guarantee, but I wouldn't be surprised to still have that lineage exactly the same way. Flippers, fluke tail, everything. It won't surprise me none. Because people forget about that stuff, right?
Well, they've got newspaper articles of them admitting shaving down a pig's tooth to make fit the fossil record, and on and on and on. And that's what that book, Bones of Contention, talks about. They have lied, and yet they say, trust us, we represent science. Well, real science doesn't lie. It follows a method. So they've been lying from the beginning for full indoctrination to what? What's the purpose of evolution? What do they want to hide?
The
Why would they
creator.
lie?
The creator. They falsely put them together. It's a false dichotomy, creation versus evolution. Well, what's evolution? Right? Has nothing to do with creation. What in the world? Right? That's the first faux pas. The second was you can't even define it. And you definitely aren't going to post the scientific theory of it. Why? Because it doesn't and it never existed. There are no experiments. There's no science in this whatsoever. None. Zero. Nada. Niente. Zippo. None. That's what the funny part of this is. Nix. 9. No way. None. 0. All right, guys. Yeah, I could go on and on with that. Let's go with final thoughts. We'll start off with 10th. Go through Adam, Paula, and then Betty will round it up.
Well, great show. I learned a lot here. Most of it I probably won't ever use again, but it's all that deep down crazy stuff they put on so that we get confused and just have to trust them because they wear a white lab coat. But things are as is. A whale is a whale. It's always been a whale. Different types of whales, but they're all whales. They're not some hyena-looking thing out of the mountains of Pakistan. So I rest my case with Adam there.
Cool. I'd just finish off with saying, or follow up on what Paula said about not necessarily remembering the detail, but the conclusion. And I think, as you've aptly demonstrated tonight, QE, that it's not so much that the detail's even there to analyse. It's omitted, and in most cases, it has to be omitted because it will undo the point they're trying to make with regards to their paradigm. So that's the thing for me that I've seen, the level of detail that's being missed in not just the genetics but the archaeology concepts as well. The way in which the story is painted with no real foundation, and in most cases, as you've alluded to, the narrative with regards to this fossil in the museum, you know, the narrative, I said it myself, the narrative will not change. Once they've put that story out there, even when it's disproved, they'll still keep it out there to convince little kids that dinosaurs are millions of years old and real, and the way they structure them. That's the real farcical thing about tonight's picture, you know. Flippers.
Flippers.
As if a dog could get flippers on. They wouldn't stay on, would they?
I don't know. Grown adults, man. Grown adults. Don't know. Paula?
Yeah, I think of all the things that disprove evolution, the complexity, the sheer magnitude of it in biology should be the number one. And yet they think that's where it's proved, in a place that most people can't go. Of course, they're going to go there, but they have to be in such deep denial when they, you know, they being the few who even try to understand it, you know, some of the people that you quoted, the PhDs and whatnot, have to be in such deep denial that this is just screaming design at every level, because to have this all work together on such a scale and to be so adaptable to account for differences in environment is something that could not ever possibly evolve, and didn't. And there's no evidence for it evolving, but they can't call it design because, as it has been from the get-go, the whole theory is predicated on the fact that they can account for the existence of anything, especially life, without a designer. And they don't want a designer. They don't want God to exist. This is all basic 101 atheism, which is really anti-theism, that makes us the masters of everything because we're the apex predator, when at the same time they'll turn around and invent space aliens who are more advanced, who seeded the planet, and around it goes. But I think the sheer complexity and the machine-like precision of these chemical compounds and so forth should be the most destructive thing against the theory of evolution. But again, it's an unfalsifiable theory because they believe in evolution no matter what. when, according to their own methods, it should have been abandoned as a theory long ago, because that's how science works. You're supposed to, if you can falsify your theory, it's not science, and if you can't falsify it at all, if there's no way you can think of to falsify it, it can't even qualify as a scientific theory. And their belief in evolution, no matter what, means it's unfalsifiable. So it just, every level you can think of, it just destroys the whole theory, and biology especially, in my opinion.
Thanks, Betty. Any final thoughts? And Edmund?
No. My voice is about to give out, so I think I'll leave it at that. And there's no admin here. But great show, and there's another one to come, I believe. Is that the conclusion?
Yeah, because of Adam, and Paula just brought up a good point about complexity, right? That's a pretty ambiguous term that they try to weasel around. You're not going to be able to do that, because next week, the final version, we're going to end with functional sequence specified complexity, right? It's going to put the friggin' final nail right through their heart. And then we're going to talk about, thanks to Adam, junk DNA, right? Check this one out, man. If you thought this was funny, wait till we get to that.
Are you dedicating that to Alan B? That section and...
Yeah, the junk DNA will be dedicated to Alan B. Yeah. What a sweetheart he is. He's not a parroter. All right, guys, a couple more admin things. First of all, I'd like to thank the panel today for their participation. I appreciate it. Betty, thank you very much.
Yeah,
Pleasure.
sure. Anytime.
Good to be here, mate.
A couple things. Ball Busters this Saturday. Make sure you guys are there. 1 p.m. U.S. Central, 7 p.m. UK. If you thought Pseudo McWestian 2 Space was a belly laugher, you ain't seen nothing yet. So make sure you're there. And the last one, as for the Christmas schedule, I didn't look forward, but somebody told me that Christmas Eve falls on a Tuesday. And so obviously New Year's Eve and stuff are falling around this day. So I'll have to take a look at it and then move from there. Maybe take a vote. Do you guys want to show? I know most of the people told me I'm not going to be around, so I don't really blame them. So let's just be flexible with that. Maybe I'll put in a couple of old videos. I don't know. I haven't looked at it yet. So just be flexible. So folks, take no prisoners. No quarter will be asked. None shall be given. And remember, I'll be back.